The topic is on brain targeted drug delivery systems. So these are the contents which I am going to discuss. It's introduction, factors affecting drug delivery to brain, barriers, what are the barriers which are related to the CNS drug delivery, physicochemical factors which affects the drug transport across triple B, approaches for brain targeted drug delivery, mechanisms of nanoparticle transport across the blood-brain barrier, pharmaceutical carriers used for brain targeting, and advantages and disadvantages of using nanoparticles for CNS targeted drug delivery. So these are the contents which I'm going to discuss here. So we know that the drug delivery to the brain, it's a process of passing some active molecules across the triple B for the purpose of treating brain related diseases. So this we can say that it's a highly complex process that has to be taken into account. Especially we should know the anatomy of the brain as well as the restrictions which are possessed or which have been imposed by the special junctions of triple B. This blood-brain barrier together with enzymes, it restricts the entry of substance for maintaining the internal milieu of the brain. So because of this presence of multiple endogenous transporters, triple B allows a selective entry of nutrients and minerals across it and limits the entry of foreign substances like drugs as well as neuropharmaceutical agents. So this is a very important concept to be considered. It allows this triple brain barrier, triple B, that is the blood-brain barrier, it allows only selective entry of nutrients. The conventional drug delivery systems, which release drug into the general circulation, fail to deliver drugs effectively to the brain and is therefore not very useful in treating certain diseases, especially in case of the CNS, like of Alzheimer's disease, dementia, Parkinson's, AIDS, viral and bacterial meningitis. So it's very when we compare the conventional drug delivery systems, seeing some newer or novel drug delivery system will be more apt in case of the brain targeted drug delivery systems. Let us see that. Why to target the drug? Especially to have an effective therapeutic effectiveness at the specific site, we target the drug. And the conventional dosage forms will have lot of toxicity, the side effects. To reduce the toxicity, we target the drug, and also to reduce the dose. So these three are the main reasons why we go for targeting the drug. So there are some factors affecting drug delivery to the brain. The first factor is triple B, that's a blood-brain barrier. Second, cerebrospinal fluid, and some of the physicochemical factors. And talking about these barriers, especially to the CNS drug delivery, this blood-brain barrier as well as a cerebrospinal fluid barrier. These are the barriers which will not allow the drug to release to the CNS. So let us see what is that blood-brain barrier. This blood-brain barrier, it's a highly selective, semi-permeable membrane that will be separating the circulated blood from the brain and also extracellular fluid in the central nervous systems. So it is this triple B. It will be separating the circulating blood as well as the fluids, the extracellular fluids which are present in the CNS. And this triple B consists of a tight junctions of capillary endothelial cells in which it restricts the passage of solutes and thus preventing the paracellular transport of molecules into the brain. It is not allowing to go into the paracellular transport. The mechanism is that it is the tight junctions of capillary endothelial cells. It will restrict the passage of the solutes and thus it is preventing to go to the paracellular transport. It also inhibits delivery of therapeutic agents to the CNS and impose obstruction for delivery of large number of drugs like antibiotics, antineoplastic agents, neuropeptides to pass through the endothelial capillaries to the brain. The lipid-soluble drugs with a molecular weight of less than 600 Daltons only can pass through this triple B by passive diffusion. So this is an important consideration here. The drugs which are of lipid-soluble and the molecular weight of less than 600 only able to pass. There are many good drugs which are supposed to be used for the brain related diseases. Since those, the important concept here is that the lipid-soluble drugs and the molecular weight less only easily it passes through the triple B by the passive diffusion. So the other drugs, it is considered as a barrier. This triple B is considered as a barrier which it is not allowing to pass the other categories of drugs. Therefore, we can say that only lipid soluble solutes The two which are easily diffusing through the capillary endothelial membrane only will pass through the triple B. So this is what the structure of that triple B. And as I mentioned, that is the tight junction. It is not allowing the drug to pass it in the paracellular transport. It has got a tightly endothelial cells. Because of this tight junction, it is not allowing the drug to pass through the to go into the next mechanism into that is the paracellular transport. Next, about the blood cerebrospinal fluid barrier. It acts as a barrier to drugs entering the CNS. Okay, not only that, it also has the drug has to reach to the CNS. So here it is formed by the tight junctions of choroid plexus epithelial cells, which are surrounded by the microvascular endothelium with intracellular gap and fenestration. These epithelial cells have an arrangement in such a manner that it prevents the entry of molecules. Only lipid soluble drugs with a molecular weight under that means in between 400 to 600 daltons can pass through this choroid plexus by the transcellular route, either by passive or active transport. So here in this blood cerebrospinal fluid barrier requirement is, ideally speaking, the only the lipid soluble drugs will cross, and the molecular weight in between 400 to 600 dalton, and that too it passes through the transcellular route, either by passive or active transport. I told that there are some physicochemical factors to effects which the drug is not passing through the why it is not going not passing the triple B, like molecular weight of the drug, lipid solubility, its partition coefficient, ionization, size and shape. There are other factors other than these physicochemical factors like cerebral blood flow, concentration gradient of drug, systemic enzymatic stability, clearance rate of the drug, sequestration by other cells, pathological status, metabolism by other tissues, affinity for reflex proteins. So these are the factors mainly it affects the drug now to transport across the triple B. So this is a transport of the molecules across the triple B. The hydrophilic molecules, how what is happening, why it is not allowing the drug to pass immediately. Whereas the lipophilic molecules, it undergoes a diffusion and thereby it is passing through the tight junction and allowing it to pass. Approaches for brain targeted drug delivery. There are many approaches to prepare the brain targeted drug delivery systems. One is the invasive. Under that, intracerebroventricular infusion, that is ICV, convection enhanced delivery, intracerebral injection or implants, or disruption of the triple B. And second method is non-invasive. Under that, chemical techniques like a prodrug. Next, colloidal techniques like of nanoparticle liposomes. Still, there are many which have been mentioned down. Biological approaches like receptor-based, cell-penetrating peptide-based, or viral vectors, and some other miscellaneous technique like intranasal delivery and endophotic delivery. So let me explain each. Invasive approach, that which is of painful. Drugs are delivered to the brain by drilling the hole in the head, and then this implant is placed in the intracerebral, or either this implant, either in the form of implant or infusion is given into the particular area intracerebral. That is, or infusion means it is given to the intracerebral ventricular. And advantage of this route is that we said in the main component that the most suitable drugs are lipophilic drugs. So here, the advantage with this approach is that a range of compounds and formulation can be considered for ICV, that is intracerebral ventricular or intracerebral administration. Here, large and small molecules can be delivered. Either it can be given alone or in various polymer formulations, thereby it achieves a sustained release. So different invasive approaches under that are intracerebroventricular infusion, convention enhanced delivery, intracerebral implants, disruption of the triple B. Intracerebroventricular infusion. It has been reported that the concentration drug in the brain is only 1 to 2% of the cerebrospinal fluid concentration. That is at about just we can say that 1 to millimeter from the surface. So the drugs could easily distributed to the surface of the brain by the intraventricular drug infusion, but it is not properly delivered to the brain parenchyma. So pharmacological effects can be seen after the ICV administration. That is, if the target receptors of the drug are located near the ependymal surface of the brain. So pharmacological effects can be seen. Limitations. The diffusion of the drug in the brain parenchyma is very low. Unless the target is close to the ventricles, it is not an efficient method of drug delivery. Example, glycopeptide and aminoglycoside are aminoglycoside antibiotics are used in the meningitis case. Second method called convection enhanced delivery. The general principle of convection enhanced delivery involves stereotatically guided insertion of a small catheter into the brain parenchyma. Through this catheter, that infusion is actually pumped in the brain parenchyma and penetrates into the interstitial space. And this infusion will be continued for several days. And the catheters are removed at the bedside. It's mainly used to deliver high molecular weight proteins. So there are some limitation with that. Like some areas of the brain are difficult to saturate fully with the infusion, particularly infiltrate tissues surrounding its cavities. So proper drug delivery depends upon the placement of catheters. But still we can say that is mainly used to deliver the high molecular weight proteins. Third method called intracerebral injection or use of implants. This by giving directly into the brain parenchymal space, like of direct injection via intrathecal catheter or controlled release matrices or microencapsulated chemicals. So the basic mechanism involved here is the diffusion, especially it is used in the treatment of Alzheimer's, Parkinson's, brain tumor, etc. Limitations: this distribution in the brain by the diffusion mechanism, it decreases exponentially with distance. The injection site has been very precisely mapped to get efficacy and overcome the problem associated with the diffusion of drugs in the brain parenchyma. So, as I mentioned that the drugs are delivered directly into the brain parenchymal cells, and the disadvantage associated with that is that injection site has been mapped correctly, or it has been targeted correctly to have a good efficacy. And fourth is the disruption of triple B. It is widely used for CNS drug delivery. That means disrupting the blood-brain barrier, like exposure to radiations and infusion of solvents such as dimethyl sulfoxide, ethanol may disrupt the blood-brain barrier. By inducing pathological conditions like of hypertension, hypoxia, or ischemia, this triple B can be disrupted. So some of the important techniques for disrupting triple B are osmotic disruption. That means osmotic shock will cause the endothelial cells to shrink. Thereby, we are disrupting the tight junction, thereby allowing the passage of drugs through that. And second is MRI guided focus ultrasound triple B disruption technique. This is the most commonly used, MRI guided. That means the ultrasound has shown to be capable of triple B disruption. The combination of microbubbles, that is perform microbubbles and ultrasound agents, the contrast or dyes, optician with a diameter of 2 to 6 micrometer, which is injected in the bloodstream before exposures to the ultrasound. This technique has shown to increase the distribution of the blood-brain barriers. Limitations, these all coming under the invasive approach. These are lately costly and requires anesthesia or hospitalization. And this may enhance tumor dissemination after successful disruption of the triple B. Neurons may be damaged permanently from unwanted blood components entering the brain. Now, non-invasive approaches. A variety of non-invasive brain targeting drug delivery systems have been investigated, making the brain blood vessel as a drug distributor. So these non-invasive techniques depends upon the drug manipulations, which include alterations like chemical techniques or colloidal techniques can be used. Chemical techniques like prodrug, colloidal techniques, nanoparticle, liposomes, microemulsion, nanospheres, and nanocapsules. Chemical techniques, that is with the help of the prodrug. Prodrug, it's a in fact is a lipid solver which can cross a triple B. And this prodrug will be metabolized within the brain and convert into the parent drug. Thereby, it is one of the technique to target to the brain. This prodrug basically it consists of a drug which are linked to an chemical moiety. This active drug is formed when this attached moiety in prodrug is cleaved by an enzymatic process. So initially it will be prodrug. Once when it is metabolized within the brain, it will convert in the form of a parent drug. Example, levodopa, GABA, valproate. These all the prodrug approach by chemical technique. Limitations of the prodrug approach are adverse pharmacokinetics, the increased molecular weight of the drug that follow from lipidation. So this is the illustration of the prodrug approach. When the drug has its moiety, it does not pass. Whereas in order to cross these biological barriers, that is blood-brain barriers, by this process called enzymatic or chemical transformation, it is able to pass without any delay, and effectively it passes. Second, the next technique under the non-invasive is the colloidal drug carrier techniques, where usually we adopt under that nanoparticles. You know that the nanoparticles, what is size, what it is made up of and all. Already I discussed in the previous sessions. It can be a polymeric materials, or it can be of solid lipids, which are in the range of 1 to 1000 nanometer. And these nanoparticle system, especially for CNS targeted, gives better penetration of both therapeutic wise as well as the diagnostic conditions, and reduce risk in comparison to conventional treatments. But by using this nanotechnology, it is possible to deliver the drug to the target tissue across the triple B, and thereby release the drug at controlled rate and avoid degradation process. Microemulsion. So we know that microemulsion is a system of water and oil, or oil and water and amphiphile, where there will be a single optically isotropic compound and thermodynamically stable lipid solutions. Having high loading capacity and good penetration ability is the advantage of this microemulsion, which can act as a colloidal drug carrier. The next is the liposomes, which we have already discussed in the previous class. These are all lipid vesicles. It will have phospholipid layers. Also, it consists of an aqueous cord for encapsulation of hydrophilic drugs, where amphiphilic and lipophilic drugs can be solubilized within the phospholipid layer. The next is nanospheres and nanocapsules. Even the nano, it can be that can be encapsulated within the nanospheres or nanocapsules, and thus facilitating its entry towards the CNS. And the drugs delivered using these nanospheres are like loperamide, tubocaine, doxorubicin, etc. Next is the biological approaches. So here it is completely based on the receptor mediated of chimeric peptides, especially coupling of non-transportable transportable peptide to a transportable peptide, where this receptor will be a mediator, and thereby it absorptive or transcytosis will happen through this TBB. Next approach under the biological is cell-penetrating peptide. It's a mediated drug delivery too. Here, it transports the molecules which are tagged within the cell membrane, thereby into the cytoplasm and the nucleus. These interact the cell surface by surface independent mechanisms. The next approach of biological is viral vectors. It has become a valuable tool, especially for the gene delivery at a specific site in the brain. It associates several problems like unwanted deleterious immune response, changes in the property of delivered virus due to endogenous recombination, and maybe due to its mutagenic behavior, which are leading to oncogenesis. Other techniques, other than these invasive, non-invasive chemical techniques, there are some other techniques too. One is intranasal drug delivery. That means the passage of drug molecules towards the brain through the nasal drug delivery. So the nasal drug delivery, nasal mucosa are used for delivering the drugs for the CNS disorders, and thereby systemic administration, especially like analgesics, sedatives, hormones, cardiovascular drugs, vaccines, hormones, especially this strategy through the intranasal drug delivery, it is successful in the treatment of Alzheimer's disease. And here the mechanism adopted will be like of to from nose to brain target. It will be of intracellular transport or extracellular transport. And the other miscellaneous is iontophoretic delivery. Let me say some advantages of intranasal. It has got rapid drug absorption, and due to the highly vascularized mucosa, nasal mucosa, it will have a rich blood supply, and thereby it reaches very fast. And drugs which cannot be absorbed orally may be delivered to the systemic circulation through this nasal delivery. It's a convenient route when compared to parenteral route in case of long-term therapy. Viability of larger drug molecules can be improved by means of absorption enhancer or other approach. Self-administration is possible. Large nasal mucosal surface area for dose absorption. Disadvantages: some of the drugs may cause irritation to the nasal mucosa. Nasal congestion due to cold or allergies may interfere with the absorption of drug. Drug delivery is expected to decrease with increasing molecular weight. Now, let me say one of the dosage form of the mechanism of nanoparticle transport across the triple B. There are usually mainly these mechanisms followed. First is like adhesion of nanoparticles to brain blood vessels. or can be a fluidization of triple B endothelium by surfactants, or by opening of tight junctions of endothelium, or by transcytosis across the brain endothelial cells, blockage of glycoprotein in the brain endothelial cells, endocytosis by the brain vessel endothelial cells. So through these mechanisms, these nanoparticles can cross a blood-brain barrier. There are some pharmaceutical carriers which are used for brain targeting, that are micelles, vesicles, nanoparticles, nanospheres, nanocapsules, nanogels, liposomes, microemulsions, dendrimers, solid lipid nanoparticles. And there are some advantages of using nanoparticles for CNS drug delivery too. CNS targeted drug delivery too. These nanoparticles protects the drugs against chemical and enzymatic degradation. Due to the smaller size, it penetrates easily into the capillaries and it is easily uptaken within the cells and allowing an efficient drug accumulation at the target sites in the brain. And especially the usage of biodegradable materials for nanoparticle preparation will help not only reaching over there, allow sustained release at the target site after injection over a period of days or even weeks. Also able to reduce side effects of some active drugs. There are some disadvantages which are related to nanoparticles to CNS targeted drug delivery. The small size and large surface area can lead to particle-particle aggregation, making physical handling of nanoparticle may be difficult in liquid and dry forms. In addition, small particle size and large surface area results in limited drug loading and burst release may happen. The cost of production is quite high. Leakage and fusion of encapsulated drug molecules. Sometimes phospholipid undergoes oxidation and hydrolysis. It has got short half-life and low solubility. So these are the aspects which are related to the brain targeted drug delivery systems.